Hi there, my name is Agape and today we're going to learn about one of the greatest feats of exploration in history, the Human Genome Project. So what exactly is the Human Genome Project? The Human Genome Project was a project that began in 1990 and ended in 2003. It was coordinated by the U.S. Department of Energy, which is also known as DOE, and the National Institute of Health. The Human Genome Project was an inward voyage of discovery led by an exceptional team of researchers that wanted to sequence and map all of the genes of humans. Because of this project, we now have the ability for the first time to read nature's complete genetic blueprint for building a human being. The Human Genome Project consisted of 3 steps. 1. Obtaining a DNA code to sequence. 2. Sequencing the DNA clone. And 3. Assembling the sequence data from multiple clones to determine overlap and establish a contiguous sequence. These clones were derived from DNA libraries made by ligating DNA fragments generated by partial restriction enzyme digestion of genomic DNA from anonymous human donors into bacterial artificial chromosome vectors, which can then be propagated in bacteria. These DNA fragments in the library vectors are mapped to chromosomal regions by screening for sequence tag sites of known sequence and chromosomal locations that can be amplified. Library clones were digested with the restriction enzyme and the sizes of the resulting DNA fragments were determined. After all of that, each clone then exhibited a DNA fragment which could then be compared to other clones to identify overlapping clones. The individual clones that are selected for DNA sequence analysis are further fragmented, and the smaller fragments are subcloned into vectors to generate a BAC-derived shotgun library. Some of the advantages of the Human Genome Project include improved diagnosis of disease, earlier detection of genetic predispositions to disease, rational drug design, gene therapy and control systems for drugs, economy boost, and more. Some of the disadvantages of the Human Genome Project include it could develop a trend in designer humans, which could then be a loss in human diversity, it could form new dangerous weapons, and it could be the foundation of genetic racism, and lastly, it could also be not available to less wealthy cultures. Despite the pros and cons, the Human Genome Project benefited biology and medicine by creating a sequence of the human genome, sequencing model organisms, developing high-throughput sequencing technologies, and examining the ethical and social issues implicit in such technology. Some of the big legal issues of the Human Genome Project are as follows: privacy and confidentiality in the way that the family or the patient should have the right to obtain data about that individual's genes, third-party interest on the way that, for example, should an employer have the right to the genetic information of an employee before employment? Genetic alteration in the way that should the law permit, encourage, or forbid the elimination of a fetus who manifests harmful genes, and many more legal issues. An ethical issue to the Human Genome Project is safety due to the possibility of off-target effects and mosaicism. Informed consent is another ethical issue because some people worry that it is impossible to obtain informed consent because the patients affected are the embryo in future generations. Social issues to the Human Genome Project based on LC are how the resulting information would be interpreted and used, who would have had access to it, and how society can prevent harm from improper use of genetic information. I personally think that if the information obtained by a successful run of the Human Genome Project was in the hands of the wrong person, it could turn into a disaster. I believe that people want to have the genome editing so that we can essentially have designer babies. With this opposition, there would be no culture and variety in the world, and this would have an effect on society. This project relates to what we learned in DNA structure and organization and in DNA replication. The Human Genome Project has to do with the research effect to determine the DNA sequence of the entire human genome. The human genome possesses several thousands of origins of replications, hence this information is essential to be able to map the genes of all humans. This project also relates to what we learned in mutation because the positive byproduct of this project was earlier detection of diseases and mutations. This means that due to this project we could be able to find out about a mutation before it even occurred in the human body. And with further enhancement of technology, maybe one day we could be able to prevent it. Did you know that there are between 30,000 and 40,000 genes of the human genome? A human being can be made from a gene count only twice as great as that of a fly or a worm. Understanding how the body works is dramatically increasing due to HGP knowledge, and most mutations occur in males. Thank you for sitting through this crash course. I hope you learned something new today, and tune in next time where we talk just a little bit more about designer babies.